the very first X-raying of a single atom consciousness in the dying brain, and half of the world's largest lakes losing water. Today, we're diving into the latest from the world of scientific research. Welcome to the Science Hangout. Our first story takes us into the realm of the incredibly tiny. Imagine capturing the first ever X-ray signature of a single atom. Scientists have accomplished this groundbreaking milestone after a painstaking 12 years of instrumentation development. X-rays have been widely used since their discovery in 1895 for medical examinations, security screenings, and scientific research. Traditional X-ray detection requires a significant quantity of material, limiting its sensitivity. However, a team led by Professor Saul Y. La of Argonne National Laboratory have now for the first time ever X-rayed just one atom by employing a specialized detector. The team selected an iron atom and a terbium atom with molecular hosts for demonstration purposes. Their innovative approach involved using a sharp metal tip positioned in close proximity to the sample to collect X-ray excited electrons. This synchrotron X-ray spectroscopy, or SX-STM, effectively identified the elemental type of materials through the unique and distinct X-ray spectra, which is triggered by photoabsorption of core-level electrons, analogous to fingerprints. This breakthrough technique allows scientists to determine the type and chemical state of a particular atom, enabling the tracing of materials down to a single atom. The impact of this discovery is, to say the least, significant, particularly in environmental and medical sciences, with the potential to revolutionize research in various areas, such as quantum information, trace element detection in environmental and medical research, advanced materials science instrumentation, and even the discovery of cures that benefit humankind. Can consciousness still exist in the dying brain? A recent study suggests that brain activity associated with consciousness may, like a fireworks show of neural activity, surge during the final moments right before the lights go out. This research has, unsurprisingly, sparked a wave of curiosity among scientists, questioning just what we experience as we're dying. This research, led by Jimo Borjigin, PhD, and her team, builds upon previous animal studies conducted nearly a decade ago. The study discovered similarities between the brain activity of dying animals and humans who have experienced a cardiac arrest. In this study, 4 patients who were under EEG monitoring in the hospital were identified for the study. With their families' consent, when life support was removed, 2 of the patients displayed an increase in heart rate and a surge of gamma wave activity, which is linked to consciousness and considered the fastest brain activity. Moreover, this activity was observed in the hot zone of the brain associated with consciousness, located at the junction of the temporal, parietal, and occipital lobes. This brain region has been linked to dreaming, visual hallucinations, and altered states of consciousness. The 2 patients had previous reports of seizures, but no seizures during the hour before their deaths. Nusha Mihailova, MD, PhD, a clinical associate professor in the Department of Neurology at the University of Michigan, collaborated with Dr. Borjigin since 2015 by collecting EEG data from deceased patients under ICU care. The other 2 patients did not display the same increase in heart rate upon removal from life support, nor did they have increased brain activity. Due to the small sample size and the inability to determine the actual experiences of the patients, the study authors caution against drawing definitive conclusions based solely on this research. They suggest that larger studies involving multiple centers with ICU patients who survive cardiac arrest and are monitored with EEG could provide more conclusive evidence regarding whether bursts of gamma activity indicate hidden consciousness near death or are a result of hypoxia, a lack of oxygen, during death, causing hallucinations and altered mental states. Imagine a world where hydrogen, the ultimate clean fuel, is produced from water without bankrupting a nation. Well, a recent breakthrough in the form of a low-cost catalyst composed of elements abundant on Earth may just have pushed us closer to that reality. It could make possible the low-cost and energy-efficient production of hydrogen for use in transportation and industrial applications. It would replace present catalysts made from expensive precious metals. Working on developing low-cost methods to produce clean hydrogen from water as a renewable energy source, a team of researchers from various institutions, led by the US Department of Energy's Argonne National Laboratory, has created a low-cost catalyst that can produce clean hydrogen through electrolysis, a process that has existed for over a century. However, the team's work follows the DOE's Hydrogen Energy Earthshot initiative, aiming to lower the cost of green hydrogen production to $1 per kilogram in a decade. Production of green hydrogen at that cost could reshape our economy. It would affect the electrical grid, manufacturing, transportation, and residential and commercial heating. Researchers from MIT and McMaster University have utilized an artificial intelligence algorithm to discover a new antibiotic that effectively kills drug-resistant bacteria. The newly identified antibiotic has the potential to combat Acinetobacter baumannii, a bacterium commonly found in hospitals that can cause severe infections. Acinetobacter baumannii is responsible for various infections, including pneumonia and meningitis, and is particularly prevalent among wounded soldiers in conflict zones. By training a machine learning model to assume the inhibitory effects of chemical compounds on A. baumannii growth, the researchers ultimately identified a promising drug candidate from a library of nearly 7,000 potential compounds. This discovery highlights the significance of AI's increasing role in the search for novel drugs, in this case antibiotics. The research, published in the journal Nature Chemical Biology, demonstrates the potential of AI as a tool to address various issues by identifying compounds with unique chemical structures. Over 50% of the world's largest lakes are experiencing water loss due to the combined effects of climate change and unsustainable human consumption, according to a groundbreaking assessment published in the journal Science. However, the study provides a new method to track lake water storage trends. By using satellite data and models, researchers from various institutions worldwide measured changes in water levels in nearly 2,000 lakes and reservoirs, which represent 95% of the Earth's total lake water storage. The findings revealed that 53% of lakes globally have experienced a decline in water storage, including well-known cases like the Aral Sea. Lakes in both dry and wet regions of the world were affected, indicating more extensive drying trends than previously recognized. Additionally, nearly two-thirds of Earth's large reservoirs experienced significant water losses, mainly due to sedimentation in older reservoirs. On a positive note, however, approximately 24% of global lakes saw increases in water storage, though, notably, this was in underpopulated areas and regions with new reservoirs. Among the lakes examined by the researchers, Lake Sevan in Armenia stands out as a positive example. Over the past 2 decades, Lake Sevan has experienced a notable rise in water storage. The authors attribute this increase to the implementation of stricter regulations on water withdrawal, particularly since the early 2000s, aimed at conserving the lake's water resources. The researchers emphasized the urgent need to incorporate human consumption, climate change, and sedimentation impacts into sustainable water resource management to address the drying of lakes, which will affect about 2 billion people. In a stark message for the aviation industry, health experts are sounding the alarm about dangerous cabin fumes that could contribute to a newly emerging disease. Led by renowned aviation health researcher and former pilot Dr. Susan Michaelis, the International Fume Events Task Force is calling for action on the toxic effects of inhaling degraded engine oil and other fluids present in aircraft air conditioning systems, and have developed a first-ever medical protocol to aid individuals affected by contamination of the aircraft cabin air supply. After conducting extensive research over 6 years, the task force, comprising 17 specialists, has produced evidence-based guidance on this matter. During every flight, aircrew and passengers are exposed to continuous low levels of engine oils and hydraulic fluids in the aircraft air supply, along with intermittent fume events characterized by noticeable levels of contaminants in the cabin. The unique medical protocol provides comprehensive instructions for both medical and non-medical airline personnel to address future exposure and events. It covers the recognition, investigation, and management of individuals suffering from the toxic effects of inhaling degraded engine oil and other fluids present in aircraft air conditioning systems. The task force research strongly indicates a connection between these fluid contaminants and the health issues experienced by affected individuals. The review emphasizes the consistent pattern of adverse effects reported by aircrew and some passengers following exposure to these fumes. These effects range from cognitive impairment to respiratory and cardiac problems, and studies have even linked these exposures to specific types of cancer. The task force's work is critical in addressing the occupational health syndrome associated with cabin air contamination, providing an invaluable tool for identifying and documenting the effects of these exposures in the future. Picture this: a tiny embryo at the 16-cell stage at a critical crossroads. Which cells will become the magnificent little being itself, and which will go on to create the all-important placenta? It turns out that the answer lies in the distribution of RNA, the molecules that hold the key to our genetic destiny. Some RNA molecules like to hang out in one part of the cell, while others prefer to take a trip to a different neighborhood. And it's this little migration frenzy that actually determines what type of cells these baby cells will become. Literally, RNA immigration decides your fate. Now, why and how does this matter, you ask? Well, understanding these cell fate decisions is crucial for unraveling the current mysteries of development, birth defects, and even finding cures through regenerative medicine. Imagine a future where we can predict and direct cell fate, paving the way for incredible stem cell-based therapies in various diseases and conditions. The groundbreaking work of Dr. Jennifer Zenker and her team is sure to be a game changer.